Καλημέρα σε όλους. Good morning, everyone. It's a pleasure to introduce Antonios Dougalis. Antonios is an electrophysiologist by training. He did his bachelor in pharmacy at the University of Sunderland. Then he did a master and a PhD in neurophysiology at the same university. And then he spent some years as a postdoc at the MRC as a career development fellow. And then moved on to the University of Manchester, where he was a senior research associate. So today he will talk to us about his work about dopaminergic neurons in the brainstem, and we're looking forward to hearing about it.
Thank you. Thank you for inviting me. Thanks for coming. I'm not accustomed to addressing audiences which are basically computational neuroscientists, I guess, or computational biologists. So I hope that I can present something which is digestible today, that's something that you can digest. So my talk today is entitled Brainstem Dopamine Neurons Activity Transitions into Sleep-Wake Cycles. And within the next 40 minutes or so, I'd like to give you a brief overview of results concerning the characterization and the functional significance of a set of dopamine neurons that reside in the brainstem rather than the midbrain. So dopamine is a very important neurotransmitter, as you probably all know, and the dopamine release is related to motor control and reward processing to explicitly linked to the circuitries of the ventral tegmental area and the substantia nigra that give rise to the nucleus striatal pathway that suffers immensely in Parkinson's disease, and to the mesolimbic pathway to the nucleus accumbens and mesocortical pathway to the prefrontal cortex. Of course, perhaps what most of the people don't know is that there are many other neuronal groups that contain dopamine in the brain, named here by the popular classification by Dastrom and Fuchs as the A groups. So that's the olfactory bulb group here, and a number of hypothalamic groups over there. The retrorubral fields denoted as the A8 group. So what is less well known is that beyond the VDA area, named the A10 area, there is a system called the A10 dorsal caudal system, which is really an extension and a continuum of dopamine neurons, further caudal meaning at the back of the brain, rostral meaning towards the nose. So the main brain, you see typical immunoreactivity of dopamine neurons in the substantia nigra, pars compacta, and the VTA, but as you move further back towards the brainstem, the substantia nigra area disappears, the retrorubral fields start appearing, and the VTA area assumes a more, if you like, midline position. And as we go even further back in the brainstem, we start finding positive neurons for dopamine co-residing with another major neurotransmitter which I'm very interested in, which is serotonin. So it's quite interesting that we have areas back here in the brainstem, especially in the dorsal raphe nucleus and the periaquaductal gray, that contain serotonin and dopamine neurons at the very same time. So I'm quite interested in these A10DC system neurons, and we only know very few things, and there are a few publications between 2004 2006 by Clifford Saper's group here, and also by Fernandez Espejo group that implicated these neurons in reward processing, nociception, and of course in sleep-wake cycles, which I'm very interested in. However, there were no physiological data or in vivo or in vitro data relating the properties of these neurons to these functions. So principally, what I aim to do during my career development fellowship at the MRC is to provide a physiological characterization of these neurons, and also to attempt to provide more physiological evidence about the functional implications of these neurons in sleep, which hopefully will be much more clear as I keep on talking to you. And the methods which are important to understand what the ways that we're doing things. And I've used a few transgenic mouse models in order to be able to identify these neurons in vitro and in vivo sometimes. It's very hard to see these neurons. They're very small and they don't have any clear cytoarchitectonic boundaries, so one needs to exploit some transgenic mouse models to see them. So I used in vitro recordings using Carnclam and Voldeslam. I labeled neurons with intracellular dyes using immunofluorescence for proton spectroscopy to ascertain the phenotype. I used Voldeslam to actually measure the kinetics of many ion conductances on these neurons and model them and try to understand the mechanism of Autorhythmicity. And finally, I used in vivo recordings and ex-vivo behaviour diagrams to shed a little bit light on the functional significance of these neurons in sleep. It won't take me too long actually to go through this, and you won't be so crazy. So dopamine neurons in the periaquaductal gray seem to reside usually around the aqueduct of Sylvius. They have a very different anatomical position from serotonin cells. Their dendrites and axonal processes usually orientate themselves towards the aqueduct. You can see that, of course, here. And there is a good overlap between these two transgenic mice that link really the dopaminergic phenotype to a green fluorescent protein. So we were quite certain that this is a good model to try to work with dopamine neurons. In terms of morphological identity, the dopamine neurons, as you can see here, are much, much smaller than serotonin neurons. They have much smaller somatic surface area. They bifurcate much less. Usually they're bipolar. And they have very, very small processes, which makes it very, very difficult to achieve, let's say, dual recordings by a somatic patch electrode or a dendritic electrode. Electrophysiologically, they also seem to have distinct properties in 5-HT neurons. These are neurochemically identified dopamine neurons or 5-HT neurons. You can see the differences in the firing rate under current ground. It's about 5 to 10 hertz firing for the DA neuron. The 5-HT neurons fire really, really slow, about 1 to 2 hertz. And also the responses to a highly polarizing pulse are very, very unique in these two neurons. This little change from the end of the start of the step, the end of the step, denotes really the activation of a high polarization activated cation current, whereas the delayed repolarization defining that you see here under Voltzkamp seen as an outward current. I will explain more about this in a minute. Is very, very important in dopamine neuron physiology, really. These are not shared by serotonin neurons, so they don't really express this kind of currents. You see the classic under break here, according to Hodgkin and Huxley, in these neurons. A very important property that is putatively related to the action of antipsychotics is the depolarization block. Dopamine neurons, when you inject more and more current into them, they go into a phase where they stop firing. This doesn't happen in serotonin neurons. People think that antipsychotics usually induce this depolarization state. I try to understand a little bit more about the depolarization block in DA neurons, but the message here is that they do enter this state, which is rather important, and rather different from the 5-HT neurons, which really they don't care how much current you apply to them. You can apply as much current as you want, and they will keep on firing and firing and firing. Usually they accelerate and then they deaccelerate, but they do not enter a depolarization block state. A physiological characterization cannot go without a synaptic analysis, really. So Voltzkamp is a really nice technique. It's something that you use because most of the conductances on a neuron, they are voltage dependent and time dependent at the same time. So what you really, really want to see is the voltage dependence of some of these conductances in order to measure the kinetics. So with Voltzkamp, by Voltzklamping a neuron, meaning keeping the voltage steady at all given times, you can look at excitatory postsynaptic currents. You can look at by stimulating the peripath depression or facilitation, which gives you meaningful identification of the processes of synaptic vehicle releasing, the vesicular release. You can look at the, if you like, the peripath rates, the amplitude and DA ratios, which are very important in discriminating, in setting up some certain thresholds into neurons. At the same time, you can do that for inhibitory potentials. So all these information are very important for neurophysiologists because we really like to kind of describe kinetically what's going on. Really, the take-home message here is that DA neurons, again, are very, very different from 5-HT neurons as far as these properties are concerned. But what I am really, really interested as well, and I guess this is a real asset to the computational neuroscientists, is the Voltzkamp technique and the way that you can use it to understand something what we call autorhythmicity. So DA neurons, even if you blocked all this synaptic traffic, you'll find that they still fire. So one of the things that we are trying to understand is how does this firing go here? So what are the mechanisms that give rise to an autorhythmicity into a neuron? So Voltzkamp will help you to actually recover particular ionic currents. Sometimes it's very easy, sometimes it's much more difficult. So sometimes you have to use pharmacology and current subtraction and some complex protocols. But the idea is to attempt to record from a single ionic conductance at any given time, and actually after recording that, trying to fit the Voltzkamp data to mostly generalized Boltzmann equations. and then give that a whole skin Huxley formalization, which I think that this is not new to you. You're all computational biologists, so whole skin Huxley should be not a new concept. And using all this together, you can pretty much create a computer model to make some predictions about how these neurons do things for you. This is a very simple case, for example. It's a kinetic analysis of a high polarization activated current. This is sensitive to a blocker called ZD7288. It's a typical Boltzmann protocol from -30 to -130 to give you this inward currents occurring here. And what we do is we calculate through current at the conductances, and we normalize conductances to the maximum conductance, so you create a steady state activation curve, if you like, here. And through that, you obviously know that you usually use these B50 values on the slope, which tells you what is the midline, if you like, or the midpoint for the activation of this conductance. And also you kinetically analyze by fitting exponential curves to the activation of these currents. So all this together can give you an indication of how this current is working. And you can formalize that in a Boltzmann-Huxley, if you like, equation or manner, and you can use that to create an in silico neuron. More complex things happen when you're looking at potassium currents, for example. These are transient currents here and sustained currents, so you can use pharmacology or two different protocols and current subtraction again to describe the activation and inactivation kinetics of these neurons, of these currents. Or you can use that for voltage-gated calcium currents. Usually voltage ramps are quite cool because they can give you the very, very small currents there. You can use two different protocols and you can subtract one protocol of the other. So in this case, for example, dopamine neurons do have two types of currents. A low voltage activated is a transient current, usually representing a T-type current. And high voltage activated, which is mnemonic insensitive, which is an L-type current. Usually if you do change the calcium extracellular levels into brain slices, you don't lose autorhythmicity in dopamine neurons, which is quite amazing, because many other neuronal processes do depend on calcium, but not this one, apparently. And finally, you can do that with calcium, with sodium channels as well. You can do slow voltage ramps, you can see persistent currents, fast currents. You can do all sorts of things with voltage down, and you can really, what I'm trying to make is the point, is you can describe all the properties of 4 ionic conductances using voltage down physiology. And that would lead you basically to creating a DA model neuron. These are just representative examples of how this model behaves really, which is pretty much amazing that it behaves a little bit like the real one. I mean, it fires at around 4.8 Hz and has a good internal ratios to the action potential. It does exhibit the polarization block, albeit at a little bit higher, if you like, gain here. It also exhibits the classic voltage sag, which is the hyperpolarization current mediated voltage sag here, and the delayed polarization. So it does replicate essentially the electrical behavior of a real neuron. And I've used the DA neuron to make all sorts of predictions about the mechanism of the polarization block. I only want to show you very briefly some simulations just showing really here the current flowing through each individual ionic channel during the interspike interval. So these are two spikes here. The action potential is truncated, of course. I've divided the phases into phase 1 and phase 2 because there's a midpoint there. Usually the first phase of the polarization depends on the delay to fire potassium current and an A-type current. But when you reach that midpoint, these currents contribute very little to the repolarization after that rating threshold. And it's usually the sodium, the transient action sodium and the persistent sodium current that do drive the repolarization firing after that. Whereas the calcium currents contribute very negatively. Here is like 1/10 of the scale of this. Of course, you can always say, I mean, this is a tiny current, it's only like 2 picoamps. How can this do something? Well, dopamine neurons have got a huge input resistance compared to, say, prefrontal cortex, which are only 100 megaohms or 200 megaohms. These guys approach 2 gigaohms. In electrophysiological terms, means that a 2 picometer current can give you 4 millivolt of deflection or even more. So if you combine two currents that give you 5 picoamps or a synaptic current that gives you 10 picoamps, then you have, wow, 20 millivolts of polarization. That's huge for a neuron. It can take it from a high polarized state to burst firing and back. And that's quite amazing with high input resistance neurons. So to really summarize, the physiological conclusions are that they do really represent dopaminergic neurons who present a different entity in the circuitry. And they have morphological, electrophysiological, and pharmacological characteristics very different from 5-HT cells. And the profile, if you like, is more like VTA neurons. So we didn't know any of these. I was expecting that I would find a new neuron, a completely different neuron, but I was unlucky. So it's not a substantial nigra like a neuron, but it is like a VTA neuron, and it does share the mechanism. The difference is that it has different anatomical connections with other groups, really. And which brings me to the functional role of these neurons. I won't bore you too much about those things. The traditional classification of any neurons that are wake active or sleep active, because I'm very interested in communications between arousal centers in the brainstem, is based on two really simple tests. One is called the C-force expression, which is just an expression of the immediate area genes. And the other is to record action potentials around the sleep-wake cycles. And if you did that to a neuron and you could find positivity in C-force expression and changes in action potential frequency, possibly you could actually claim that it's a wake active neuron. What we know already about dopamine neurons is that they do change the sleep architecture by reducing actually the amount of wakefulness. And they do also express C-force activity, and also they have some reciprocal corrections with many wake active nuclei. But we don't have any data about how do these neurons change their activity during sleep, or what are the mechanisms of these changes, if there are any. Well, to do that, I have undertaken some in vivo recordings. I prefer the term active rather than up and down. These are neurochemically identified neurons here for dopamine. This is the position, the action potential, and simultaneous recording from a DA neuron. These are actually data that my colleague Judith Weimer managed to actually get. It's very hard to record these neurons. There's about a handful of neurons recorded within 5 years, and no one else has ever managed to actually record them. And real dopamine neuron, that means neurochemically identified dopamine neuron in vivo.
Why is that? Why so difficult?
There is no clear cytoarchitectonic boundary, so you really don't know where you should be going. So you can record from some cell, and it's also very challenging to label the cell via the cellular technique. But the most important, the problem that we're faced is that you really don't know what neurons you're recording. So with the advances of science now, we're trying to record from these neurons and also label them so we can understand a little bit better the secretary and the connections. So what these data have shown, Judith was very, very lucky to get these neurons, and she showed that really these neurons fire at the up state, or like active state, because up state denotes intercellular potential of the EEG, denoting that probably they are related to sleep and wakefulness, and probably they are wake active because we would expect them to fire at that particular time point. But it's very hard to stimulate because you're probably going to lose a recording. So it's very hard to make any further inferences from in vivo data, purely because you don't have really the technical aptitude to actually perform such recordings, unless you wanted to spend, let's say, a few years creating an optogenetic model, whereby you can lower a fiber optic and activate these neurons. Traditional physiological techniques with stimulation would not help you a lot to see really whether stimulating this neuron would cause electrocortical arousal. So to resolve this, I've taken an alternative approach, and I've used a brain slice preparation, which I guess most of the people consider as being rather inert, or rather severely severed, severely damaged. But it's not always the case, because we already know that the neurons like the neurons in the suprachiasmatic nucleus, they do exhibit differences in environment in vitro, something that no one had expected before. So what is the important thing about the in vitro slice is that neurons do maintain some of their properties. And unlike what people think as that being a grossly damaged preparation, they do actually do certain things like in vivo. So one approach to look whether these neurons are wake active is by recording from brain slices that have been obtained at different times of the day, or applying forced wakefulness or sleep-inducing protocols, ex-vivo, that means in vivo, and then splicing at different times of the day and looking at the activity of these neurons. And in fact, this particular procedure has proven very useful in my case here, because I found that by actually cutting slices at different times of the day and recording from these neurons, not only changes the likelihood of finding a DA neuron spontaneously active, but also increases the frequency of these neurons. And these properties are not shared by the EPA or the substance in neurons, which means that they are more specific to the dorsal raphe circuit rather than the dopamine phenotype. Of course, one of the most important questions that we'd like to ask is what drives this diurnal rhythm? I mean, you can see that in the daytime they're firing less, which is well in the biphasic model of sleep of mice, which means that when there is light, they sleep, and when there is no light, when it's dark period, then they move around. But what is the drive behind this rhythm? My favorite candidate were potassium currents, but despite the fact that we got a difference, change in the input resistance, I will explain very shortly what this means. None of the potassium channels seem to be changing during these sleep-wake cycles, so it really means that potassium conductance do not seem to account for these firing differences, but surely a change in the input resistance, at least from a physiological point of view, means implicating a reduction in the conductance. So I'm very interested in chloride conductances because basically they are used by GABAergic and glycinergic mechanisms. And I tried to see whether there is any difference for the chloride potential, and I found that yes, indeed, there is a change in the amplitude of spontaneous IPSCs arriving at the neuron. And when you try to find the reversal potential, you find that actually these neurons do have about 15 millivolts or 10 millivolts, if you like, sometimes, of difference between day and night, which might explain the differences in the firing rate from day to night. So to explore a little bit more about the synapses and how do these synapses change and how do they contribute to the changes that we see in these neurons, I recorded images with IPSCs, which are preventively famous this time, on the spontaneous IPSCs, and found out that, in fact, that when you actually record IPSCs, inhibitory post-synaptic currents, after a long period of sleep, you seem to have weakened the inhibitory synaptic efficiency, and that materializes a change both in frequency and in the amplitude rate of these events. The non-stationary fluctuation analysis, which is a nice trick that we like to do in electrophysiology, is trying to extract some information about how many channels are active at the peak of the current and what is the single channel conductance, if you like, of this current, suggests that there is a reduction in the conductance of the GABA current. Of course, there are changes in the propensity of bursting. There are also changes on the temporal accumulation, shown here by peripass ratios here. So in the daytime, when they are mostly sleeping, and after a period of sleep, the peripass, if you like, facilitation that allows for temporal summation of GABAergic machinery turns into a peripass depression. As expected, if you load the neurons with high chloride, you will no longer see these changes in the amplitude, but you will see the changes in the frequency, suggesting that there are some presynaptic changes occurring on dopamine neurons. You can ask at any time if you want, please interrupt.
I think maybe you will.
Sure.
I may have missed that.
Yes.
But in view ex-vivo, is the rhythm retained the same way that happens in vivo?
Yes, go on.
So it doesn't have to do with dark, light, sleep, or whatever?
No.
So it's
Sleep and wake denotes the time that the slices were taken according to the history of the animal.
Yeah, but how long can you maintain the ex-vivo situation of whatever?
The data suggests that once you cut slices at a particular time, for these particular neurons, they seem to be trapped into the state that they were prior to cutting the slices. Yes. So that's the important thing about them.
So that's synchronous for the whole population of cells in the slides?
It's quite synchronous, yes. It's quite amazingly synchronous, and when you record IPSCs and EPSCs, the very same, from the very same cell, because these are simultaneous IPSCs and EPSCs by exploding different potential, you actually see very good EBSs in the daytime and very large IBSs in the daytime, and then the reverse happening as they move along. But yeah, they seem to be trapped in a state where it reflects the prior history to what was been happening in the animal. The important thing is that, as I said, is that this is also happening on the reverse order for excitatory synapses. So when you look at periods of sleep towards wakefulness, we have a strengthening of excitatory synaptic frequency. There are not changes in the amplitude. Of course, again, the peripath depression is much bigger or much less pronounced as we go to the wakefulness period, suggesting again there are some presynaptic changes in the excitatory or dopaminergic machinery. Yeah.
So when you say presynaptic changes, you mean some kind of trafficking of receptors, or what exactly would be the mechanism?
Presynaptic changes usually are
maintained in a slice.
Presynaptic changes are usually related to the probability of neurotransmitter release. Perhaps it's related to the machinery of the release of the presynaptic terminal. So it could be anything if you like. It may have to do with proteins that help the fusion, the vesicular fusion, if you like, the presynaptic membrane. Perhaps it would be more clear to understand that you're very right to say that certain interpretations are very hard, even when you talk about presynaptic changes. And a parsimonious explanation would be that we have unsilencing of some receptors. For example, lots of LTP mechanisms occur by having gamma receptors where they're not working, and then all of a sudden they do start working. And that is registered as an increase in the firing in the frequency of the presynaptic terminal, but it's not really a per se change of the presynaptic frequency. Actually, it's a postsynaptic change. But these are the caveats within which someone must work. And in order to clarify further, one should try to ideally do optical imaging. But in general terms, when you do see changes in the presynaptic terminal, you think that these are related to the machinery that causes the vesicular release. That is, the probability of release has changed. And the general understanding of these reciprocal changes, if you like, can be, if you like, summarized in this model. So we find that there are changes in the firing frequency on these 5 different points. Black is when lights are off, lights on is the white bit here. So this is the time where they are awake, and this time they're awake. This is the time when the mice are asleep. So what we find is that there's an increase in the frequency from the early day to the late day, and there are also changes into the GABAergic machinery and the excitatory and dopaminergic machinery, suggesting that perhaps there is a correlation between the early period of sleep where there is much inhibition and very little excitation, and that causes the neuron not to fire a lot. But then, as we move along after periods of sleep, we find that the excitatory machinery has been strengthened, and the inhibitory machinery has been depotentiated, and that causes the neuron to fire more. And then it follows that reverse cycle from time to time. So there's a self-limiting oscillator, if you like, if you were to consider this kind of changes occurring on a synaptic level. But surely to understand how these guys are wake active, from a simple physiology point of view, you need to prove 2 things. You need to prove that forced wakefulness means that you keep a mouse awake for a while, and you compare it to mice that were sleeping. They can find changes in the firing rhythms of these neurons. And also the reverse, that you can't find any changes when you do a forced sleep paradigm. That means injecting them with hypnotic drugs and looking at whether changes occur on the synaptic level and on the firing level altogether. So when you keep a mouse awake and compare it at the same circadian time with mice that have been sleeping, I find that there's a major increase in firing frequency, and the number of active neurons now changes. And of course, there's a change in the holding current, which means basically the magnetic potential. But these changes are not dependent, again, on potassium current, which is amazing, because I would expect that would be the case. But what are they really dependent is on pretty much excitatory transmission. I find consistently that there is an increase. Actually, according to non-stationary fluctuation analysis, we make the inference that there is a recruitment of AMPA subunits in the synapse, and that is what is causing this maintenance of this depolarized state post-force wakefulness. The GABA are similar, does not seem to change. What we get all the time is an increase in the amplitude, so you deem that as being a postsynaptic change, and that's what all these techniques of noise analysis give you an indication of things like, are there more subunits in the synapse, or is it the single channel conductance that has changed? Of course, you can always do something like immunofluorescence and find out whether the quantitative immunofluorescence find out there's more subunit, more receptors in that synapse. But in this case, the data suggests that there are increases in the number of channels, that are there in these synapses, suggesting that during post-wakefulness, we can actually increase the frequency of these neurons, and that would be dependent, at least, in a potentiation of the Dopaminergic neurons. Yeah.
So this means that essentially you have something similar to LTP when you have insertion of.
That's correct. Yeah.
But on a time course, which is, I don't know. So you have coming in and going out receptors during the day. Right? This is not dependent on prior experience or learning or anything.
No.
Has this been described before?
No.
And how, what would you think would cause this trafficking to be dependent on the cycle, the sleep cycle?
We believe that these are depending on fundamental mechanisms of homeostatic synaptic scaling. That's which I will get to. It is basically the firing of the neuron is actually there's a type of plasticity called homosynaptic plasticity, if you like, and one of the ideas behind that is called synaptic scaling. Is that you mentioned LTP very correctly. Cortical neurons accumulate excitatory synapses all the time, but if they run out, the more they have, the more excitatory synapses they have, the more they fire, and that can get out of control. So the neurons utilize mechanisms by which they detect their own firing and then they modify the synapses accordingly. So they actually reduce the input that they are receiving. And in fact, sleep itself can be a homeostatic mechanism, because that is what we believe the function is. The function of sleep is to renormalize synapses. The difference is that these neurons, which are supposed to be wake active and actually impinging on electrocortical arousal, the synapses that are impinging upon them are changing all the time. And that is quite amazing, because they produce a kind of a set-limiting system by which the more dry they get, the more they fire, so the more they excite the, if you like, the cortex. And then at some point they start reversing this order and they start becoming much more silent, allowing thus for other states of consciousness to arise. The general understanding this level building.
I was just wondering if there's the same kind of mechanism.
Yes.
For this homeostatic plasticity versus the classical LTP or LTD plasticity, what would be the difference between them? I mean, how does the synapse know that this receptor should be stabilized or turned over, or it doesn't really matter? I mean, it could be the same players.
I don't think it really does matter, because within day and night we don't find any differences in the amount of AMPA receptors that are located in the synapses. It's only the presynaptic frequency that we find, and also the firing rate. So the amplitude changes and the insertion of AMPA synapses seems to be on demand changes. So even if I let you sleep deprived for about 2 days, and then you go to sleep, I can still wake you up if there's a fire in the room, and that, in the mechanistic approach of sleep physiology, we call it an allostatic mechanism. That means on-demand alertness. So you have the circadian, which is an endogenous rhythm, the homeostatic, which relates you to the sleep pressure, the need to sleep, and then you have the allostatic mechanisms, which are on-demand alertness. So these changes in the postsynaptic mechanism denote that perhaps there are allostatic changes, that the amplitude of the excitatory Dopaminergic famous relates more to an allostatic or codes for an allostatic change.
Excuse me, but if I understand well, you think that the dopamine neurons may regulate the excitatory synapse as a postsynaptic target?
So these are excitatory.
Dopaminergic neurons.
These are excitatory synapses formed upon dopamine.
Upon dopamine.
Upon.
So a signal from dopamine neurons might act as a retrograde signal to the excitatory neuron towards them.
I'm not quite making that claim, but go on. I'm interested to
Because
see.
if the dopamine neuron gets some input, an IPSP or an APSP signal, right, there,
Yes. Yeah.
depending on whether it's a wake or sleep
Yes.
condition, then somehow, and since the APA receptors do not change in numbers, as you say, then somehow this excitation that the dopamine neuron gets must stop, right? So is it possible that the dopamine neuron, when it's active or when it's getting another signal, a circadian signal, goes back to the excitatory neuron, the glutamate neuron, and
It could be
decreases the signal it gets, right? I don't understand how
Whether
the dopamine neuron is getting excited or not excited, depending on the cycle, on the condition.
Yes, I mean the excitatory, the glutamatergic terminals that in principle then can be coming from brainstem areas that are important in REM sleep or wakefulness. So we know that there are a few excitatory connections that are in the brainstem area. So them themselves can be providing the excitatory signal for the neuron. So that there are these two possibilities that I was about to go through later on, debating on to explain the differences. One is that these changes are externally driven. So whatever happens to dopamine neurons, it depends really on terminals that reside elsewhere. So that is one extreme case. So they are just puppets. So they are transformed, their activity is transformed due to the activity of other neurons elsewhere. The second potential explanation is that there is a crosstalk between their firing activity and the actual changes that occur on synaptic level. And I'm in favor of the second because we see reciprocal changes that are very, very difficult to explain by having two different groups of neurons, inhibitory and excitatory, actually doing this very, very same thing all the time. So this is something that one needs to investigate further. Of course, it's an extra thing, and I wasn't intending to go into that. What I did intend to show you is that you can also get, when you do forced sleepfulness experiments, you can also get a depotentiation of the excitatory transmission. And actually, this is by actually reducing the amount of ampersand units that are contributing to the peak of the EPSPs, suggesting that when mice are sleeping, then we have a depotentiation of this signal, suggesting altogether that this could be a mechanism by which dopamine neurons utilize to actually reach differential on any target that they've got. Really, the summary is that these neurons do fire at the active phase of the EEG, and they do have a diurnal variation of firing activity. It is dependent on sleep-wake history based on analysis of circadian times and times of slicing, which I haven't presented to you here. I find that during periods of sleep, the excitatory drive is strengthened and the inhibitory drive is suppressed, which is in good, if you like, in good agreement with the understanding of what should be happening on a wake active neuron. The periods of forced wakefulness suggest that these neurons are truly these neurons are truly wake active, and we find also that the forced wakefulness and the forced sleepfulness experiments also most importantly is that they do not change potassium conductances or other conductances on these neurons, but they are changing consistently the balance of synaptic inhibition and synaptic excitation, which is something that hasn't been reported before, at least at the very same time. And I guess one of the physiological implications that all of you probably asked about that is that we know that the likelihood of being asleep is controlled by two processes back in '84, but now we think that there's a third process. So there's a circadian process encoding for an oscillation, biological internal oscillation. There's a homeostatic process that relates to the history, which is what we call the sleep pressure or the need to sleep. And there's a third process called allostatic process, demanding encoding for on-demand alertness. So one of your questions that I haven't answered, because I would have to speak for another 40 minutes and present you more and more data, is are these changes circadian or homeostatic or allostatic? So quick answer to that is that I find that the frequency of the excitatory postsynaptic process is inversely related to the homeostatic factor. I find that the GABAergic machinery is got mixed properties of allostatic and circadian changes accounting there, and the postsynaptic changes that I find that the dopaminergic terminals are mostly allostatic. So it's a very, very fine interplay. What is important is that there are consistent changes taking place, not on the intrinsic excitability via ion channels, but really on the synaptic fields, on the balance of excitation and inhibition. One of the most important physiological implications, I guess, which again we talked a minute ago, is what drives the expression of this diurnal rhythm in synaptic efficiency. And one would be that everything is externally driven. So other cells drive these cells, and these cells drive other cells. Okay, so these are patterns. But also, the other extreme would be that there is an internally driven firing activity modifying the synapses themselves. And that's what we call synaptic scaling. It's a very, very fine mechanism that occurs in cortical neurons and hippocampal neurons. So the truth may lie between these two extremes, but it does raise the intriguing possibility that sleep homeostasis is expressed on a cellular level by mechanisms of homeostatic synaptoplasticity. And I'll leave you to that thought, really. Last but not least, the unsung heroes are Mank and Kazuto for getting me these fantastic present lines. Without that, I wouldn't have been able to target dopamine neurons. They were very hard to find. Judith Weimer for her immense help with in vivo recordings, and as I said, she was the one who was successful in labeling those DA neurons and getting simultaneous electrocortical recordings and single-unit recordings. And of course, Mark Angel is a medical research council for the Career Development Fellowship. This work was funded by the Medical Research Council. And thank you for your attention and your time.
How clear is the distinction between sleep, homeostatic, and allostatic processes?
Not very clear. Not very clear. But homeostasis usually denotes the fact that on a clinical level, homeostasis is dependent on how much slow wave activity you have on the cortex. So if you didn't sleep for a few hours, your EEG will become desynchronized. So to desynchronize, it will become synchronized. So you will get slow wave activity appearing on your EEG on the cortex. That means that basically sleep is invading your reality slowly. And that is the way that we determine sleep pressure. So from a clinical level, sleep pressure is related to how much slow wave activity you will have on your cortex. So the more sleep deprived you become, the more slow wave activity you will find on your cortex. So when we're talking about sleep pressure, in all sleep physiology methodology, what people do is that they need mice or sleep deprived. So that increases the need to sleep. However, it doesn't change the circadian factor, which is an endogenous rhythm, 24 hours, dependent on the light. Even without light, is still circulating on a 24-hour.
For how long?
Forever. In for the suprachiasmatic nucleus, it doesn't really matter. Yes, it uses about 15 minutes a day, so it will get sicked. But it will be, if you put the mice in total darkness, they will keep on oscillating on a 24-hour basis, silent, active. So you have this circadian rhythm, and you have a homeostatic rhythm. At the very same time, according to Saker, you have an allostatic process, which is exactly what you said. I believe it's an LDP-like process. But I don't think that it will last merely so long as an LDP, because it's on-demand alertness, really. I find that the excitatory terminals do seem to suggest that there are allostatic changes occurring on them. So whenever you wake up a mouse and you compare it to mice that have been sleeping, you find there is an LDP-like change in the amplitude. However, what is very interesting is that the amplitude and the frequency of the dopaminergic terminals disengages at some point. So you can see clearly, I haven't shown you all this data because it would take me, I mean, you asked all these questions, fantastic. But it would take me at least 45 minutes more to give you more data and explain and convince you why is what I'm saying is what I think is the right thing to say. So to answer your question is that there are no clear boundaries, but you can actually approach these ideas. The the research is based upon the idea is to understand what is the expression of the need to sleep on a synaptic level. Because people do look on the cortex all the time, it's cortical neurons all the time, but they never really look on arousal neurons and see how these guys are getting modified by their synapses and how they actually change their behavior from day to night. So usually mechanisms are just, the mechanisms of arousal neurons, they just say to you, oh, well, they fire more at day when they are awake, and sorry, at night when the mice are awake, and at day when they're sleeping, they fire and relax. But no one ever tells you why do they do that. Up to now, what we knew it was that principally it was the potassium currents that they were changing, especially in the suprachiasmatic nucleus, also some chloride conductivity change. But concerted changes in synaptic potentials have never been described.
I'd like to ask, could this pattern shift be the cause of a negative feedback loop that acts more in a more quantitative manner and outside the synaptic force? That will explain how the plasticity observed is due to a different kind of machinery and not due to the changes in the neurons themselves.
What do you propose would be this negative feedback? What would be the form of it? What kind of mechanism would you envision that such a negative feedback?
I envision a mechanism that could identify changes,
yes,
but not be part of them, and would act more independent.
I see. Well, part of it is synaptic scaling. It is all about that. What you exactly said is about, but it's an integral part of the neuron. We don't really know exactly how it happens, but we know that the neurons seem to be sensing their firing activity, and they're modifying synapses accordingly. So yeah, if you were to call that negative feedback, yeah, that's my favorite theory as well. I haven't proven that anything like that happens. Principally because the tools to do that in vitro are so gross that even if you did it, it wouldn't really amount to something that I would deem as scientific revelation. So you can actually do some experiments like Diurnal does. This is a very Diurnal is a very key player in synaptic scaling. So they do experiments with TTX and they block the activity and they see changes. But these are all in cultures and their protocols are gross, really. So even if you did it, you may have a good journal in your science or Nature paper, but I don't think that the scientific significance would be there. So what we believe is that using sleep itself as the modifying procedure to elicit synaptic changes is much more robust and much more physiological than actually utilizing other drugs or things like that. So that's what is the key point that I found out through doing that. Because I started with physiological characterization and started to try to understand the mechanism. I didn't even know whether these guys would do anything like that. It ended up showing really, really consistent changes in the machinery. So it's quite amazing that you get ideas of what could be happening in the central nervous system by your own project.
There is a puzzling thing there.
Yes.
Which is why the rhythm has been synchronized according to daily life.
Why?
There's no explanation for it.
Yes.
There's a rhythm, and you have to sleep every 6 hours or 10 hours. What does it have to do with whether there is light or not?
Well, what we saw
and
is
actually, the second part of that is what happens to animals who don't share the same activity-related, day-night activity-related things with mice. But mice, usually you keep them under what? Artificial conditions, 12 hours light, 12 hours night, and they sleep during day, or what?
That's correct. They are day, yeah.
So what happens to animals that they have the reverse activity, whatever, habits?
To answer your first part of the question, the light is not important for these neurons. So even if you
timing is the thing.
Well, time,
not light. It is not.
No, but in circadian physiology, sleep physiology, the circadian time is related to light. We call it Zeitgeber, which is basically the time that the light gives you time zero. So these neurons have got nothing to do with light. They don't even receive inputs from the retina or anything like that. what we think is that they are coding for the history
And
of the
how is
animal.
it again? 24 hours?
I believe what?
So the period, the full cycle is 24 hours?
Yes. 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 So they're not related to light, and I have not data to
It's
show you.
related to the to the way the Earth moves.
That's correct, because the periaquaductal nucleus has evolved, in fact, to actually get the signal from the retina. It's the only place in the brain with the hypothalamic nucleus that gets a direct input from the retina.
Yeah,
and
my
synchronized.
question is, what is the biological origin of this thing? I mean,
The biological origin.
Yeah.
Evolution.
it's
Okay,
evolution of what?
You have
Of what?
the function is that you have.
The period.
Yes. The function is that you have a number of.
Perhaps has to do with nutrition, or else. I mean, something else, not really with the day and night, or
That also.
Yes, it could be. The function generally is that you have two systems. You have the circadian system, which is in the hypothalamus, and you have many brainstem systems that are wake active. You have the locus coeruleus, the neurogenetic locus coeruleus, the cholinergic pedicle-condyne tegmentum, and you have the dopamine neurons in that area, the serotonin neurons. So all of them seem to be wake active, and some GABAergic neurons are sleep active. There are other neurons in the ventrolateral preoptic area that are sleep active. So there are many centers, but the ascending reticular system from the brainstem impinges on the cortical arousal is so far among the body. So experiments where you actually in vivo you actually blood stimulating electrode and you inject some current and you actually cause electrocortical arousal have been done a couple of times back in the 60s and 70s. So there are two systems in the brain really, an arousal system that is in the brainstem, if you like, and a circadian system that's in the hypothalamus, and it also affects hormones. What is important is that we did not know the mechanism behind changes from sleep to wake, and this data suggests that are independent on changes on the intrinsic excitability itself so much. They're more dependent on the values of excitation to inhibition. So as far as I'm concerned, these neurons can actually receive inputs from the spinal cord, so they can be, if you like, reporting and coding in internal state to the animal. So it really depends on where they get their inputs. Usually they get them from the brainstem, but this is what they do. You know, you have a heavy meal and then all of a sudden your sleep-wake behavior changes. So this is what these guys encode, and I think they are encoding that through bias inhibition and excitation.
What about the other thing? About animals that get the reverse?
The reverse cycle. I haven't tried the reverse cycle, but it wouldn't really prove anything because they would still be having the same. If you were to do that, you would expect that. If you were to put them in a reverse cycle, you would expect that the date in our daytime, they would do basically the reverse behavior. I have never done this experiment, but I've checked in a city cycle, and I found that they are also where they should be expected to be. So I'm pretty confident that they are related to the history and rather than the circadian time.
Thank you.
Thanks for your questions.
Okay.
Thank you.
Thank you.